In April 2018, a team of researchers gathered from the University of Western Australia conducted a study in Princess Royal Harbour of Albany, where the status of invasive marine species and their impact on the biodiversity of the Princess Royal Harbour water body was investigated. The study aimed to survey and record the presence and distribution of invasive marine species on man-made structures, which act as suitable settlement points for invasive larvae. This was completed at 8 locations in Princess Royal Harbour, with each site being located at varying proximity to the Port of Albany woodship terminal, the defined point of introduction of invasive species. It was hypothesised that the presence and abundance of invasive marine species in Princess Royal Harbour would increase with increasing proximity to the Port of Albany. The study sites were the Port of Albany, including the woodship terminal and berth 3, the Albany Marina, the Albany Town Jetty, the Princess Royal Sailing Club, Shane's 2 Shipwreck, the Camp Quarrenup Jetty, and the Camp Quarrenup Isolation Jetty. 2 sampling techniques were used to assess the connectivity of invertebrate larvae between the sites and the distribution of invasive sessile species. Settlement plate sets were submerged at a depth of 1 metre from man-made structures in position to be exposed to maximum current variation on February 12, 2018 at 6 locations. 5 plates were positioned on a metal star picket pole with cable ties in an alternating sequence of hardy flex and polyvinyl chloride. At the port sites, 10 plates were attached to each pole. The varying plate types were used to test for settlement surface suitability. The plates also had dissimilar measurements, with the HardyFlex plates measuring 15 cm by 15 cm and the PVC plates measuring 10 cm by 10 cm. After 2 months, the plates were retrieved between the dates of 6th to 9th of April 2018 by the researchers. For the second technique, pairs of snorkelers were deployed to sites with a surface support team present. 5 10 by 10 cm scrapings were collected haphazardly at a depth of 1 metre from the sites. The Port of Albany sites were excluded from scrape sampling due to occupational health and safety rules. Substrate surfaces were variable, with wooden pylons being the substrate at Camp Quarrenup Jetty, Isolation Jetty, Yacht Club and Town Jetty, polyethylene at the marina, and rusty steel at the Shane's Shipwreck site. The sample was taken with a metal quadrant with paint scraping tools being used to remove the substrate material. The plates and scrapes were individually placed into labelled Ziploc bags and taken back to the Camp Quarrenup temporary lab for analysis on the same day of retrieval. GPS coordinates and site descriptions were also taken for sites, with notes on overall weather conditions being noted. Species identification was undertaken using compound microscopes, guides, and textbooks. For the settlement plates, species were identified and their abundance was recorded. For the scrapings, the presence and absence of invasive species was recorded. We did not find a significant relationship between distance from the port sites and richness of invasive species. This led us to reject our hypothesis. The sites with the highest invasive species richness were the Marina and the Yacht Club. Although the plates had significant similarity within sites, there was a lack of connectivity between different sites. This was even more evident when looking at the scrapings. However, there were some species that had significant presences amongst all sites, including the striped barnacle, Spirorbidae, and Serpulidae. These species properly illustrate the impressive characteristics of invasive species through their ability to colonize, invade, and thrive in environments. Many factors could have led to the rejection of our hypothesis. Hydrodynamic processes such as currents could have played a role in the dispersion of the invasive species. There is also high traffic of recreational vessels within the Princess Royal Harbour, which could have been used by invasive species to expand their ranges. Some sites in Princess Royal Harbour may have also had a more suitable environment for the development of certain life stages of the invasive species, such as larvae which had been freshly dislodged from the accumulation of organisms attached to boats. It is important to note that invasive species often have very different dispersal methods and could have skewed our results. Much higher abundances were noted for HardieFlex plates when compared with the PVC plates. This could be the result of the amount of time given for species to settle on the plates, as well as the species' settlement substrate preference. HardieFlex is cement-based, which has been shown to be a more suitable substrate for the species that have been identified. The skeleton shrimp was the only species which had higher abundances on PVC, exhibiting further that species preferences are key. Plates were also affected by current, with the plate side exposed to current having visibly higher species count. In future studies, increasing sample size and considering additional abiotic and biotic factors could help reveal the relationship between invasive species richness and distance from the Port of Albany more accurately. From this study, we conclude that an increase in the distance from the Port of Albany did not yield an increase in species abundance and invasive species richness, and therefore rejected our hypothesis. The drivers behind these results, however, still remain unclear. Whether it be the result of currents, the trajectories of vessels within the Princess Royal Harbour, the various dispersibilities of the invasive species, or some other alien element, one thing remains clear: these invasives must be stopped at all costs.